My name is Jennifer McManus and I'm the Head of Physics at the University of Bristol. So we're interested in protein self-assembly, and we're interested in how and why proteins associate with each other in different contexts. So one of those contexts is in biomolecular condensation. So why in cells or in the body, proteins come together to form segregated liquid phases, which can perform biochemical functions. We also are interested in protein formulation, so in stabilizing protein-based drugs so that they're safe for delivery to patients, ideally in their home. And we work quite a lot on looking at the effect of excipients on industrially relevant proteins. We're also interested in developing new techniques for looking at protein assembly, again, in everything from live cells to in vitro in the lab. So CBER DSF is a differential scanning fluorimeter, and it helps us to assess aspects of the structural stability of proteins. So we use it primarily in the work where we're formulating proteins. So there's many challenges in looking at protein assembly, particularly when those proteins are industrially relevant, going to be used for protein-based drugs. Often they have to be formulated either at very, very low concentrations or very high concentrations, and the analytical tools available to do that are quite limited. The really good thing about the DSF is it allows you to make measurements across that concentration range, which helps us to formulate protein-based drugs that can be formulated at concentrations which allow small volume deliveries to patients, which has a real-world impact in their quality of life during treatment with biologics. So I would call the DSF, the Super DSF, convenient. For me as an academic, it has been a fantastic tool in the lab. It has been really useful for project students, for example, to generate large amounts of data in a relatively short time during their project, but we've also employed it in our research activity for PhD students and postdocs. So it's been a very convenient tool. So we've been working with Protein Stable as part of a research project, and they've been really responsive and interactive in that time. So we've sort of worked collaboratively to use the DSF for our needs here in the lab, and Protein Stable have been really helpful in helping us create a tool in the lab that meets our needs.